I am David Koslicki. I've recently moved from a mathematics department at Oregon State University to a computer science and engineering and biology department at Penn State. So what I'm going to be talking about today is how we might be able to use large biomedical repositories to find new uses for already FDA-approved drugs, which is a little bit different than the stuff that I normally do. Usually I work in metagenomics and sort of mathematical computational biology. But for today, the outline of the talk is as follows. We're going to go over what motivates this problem, integrating biomedical repositories. We talk about large-scale effort to be able to use all this information that exists called Translator. We'll try to integrate all this information into a thing called a knowledge graph, learn on those graphs, and figure out how we might be able to do drug repurposing by using that information. So let's go ahead and begin in the very beginning and motivate the following problem. So we've already seen this a little bit mentioned in some of the previous talks, but just to recapitulate it, let's go a little bit meta. Where does the information that you gather end up? So where does the knowledge you generate go? For example, let's say that you find that a certain gene mutation contributes to a disease, and this has been computationally or experimentally verified, and it is now fact. Well, this could get picked up by a couple of different repositories of info. So for instance, the Online Mendelian Inheritance in Man repository, or OMIM, might be one of these that picks that up and says, okay, we'll record this bit of information. You could find that a certain non-coding RNA regulates a particular gene or the expression of a particular gene. That might get picked up by another repository called Mirgate, and that will record that little fact. Or you might be doing protein-protein interaction networks, and that could get picked up by Pathway Commons or Reactome, etc. So there are lots of different repositories of info out there, as I am sure everybody here is aware of. We got things like KEGG, which has proteins and protein families and pathways, etc., gene ontology, and a whole bunch of other, that didn't work, a whole bunch of other repositories that are out there. This is the first time presenting on my iPad, mainly because my laser pointer doesn't work on this thing. But if you press and hold, you get a little on-the-screen laser pointer. So we'll see how this works, but we may be doing some zooming back and forth. But in any case, so a couple people already mentioned this, or actually mentioned this as well, that these database sizes are growing. So for instance, if you look at gene ontology and you look at all the different unique Go terms in there, you can see that this is increasing in size over time. Some of the repositories are growing even faster than others. So Chembl, for instance, stores chemical information and drug information, and this is growing. You can see that we have millions of unique compounds inside of Chembl, and they're all dwarfed by the number of unique entries in UniPro as well. So you can see that this database is just exploding in size. So since we have all these different repositories and they have a ton of different information that is increasing in size as time goes on, how might we be able to utilize this information to discover new facts in biology and in computational biology when it's not possible for any individual person to be able to go and integrate this information manually? So databases are growing. There's lots of them. They have a lot of information in it. Unfortunately, one thing that makes this kind of task difficult is that oftentimes these repositories are siloed. In other words, that there is little interaction between these repositories. This, of course, is being mediated as time goes on. But for example, let's say that you have a simple kind of question. You want to ask what metabolites interact with a gene that causes a mutation, or in which a mutation causes a disease that has a certain symptom or phenotype of interest. So a perfectly reasonable kind of question. I don't know, I just came up with it off the top of my head. But in any case, you can see that we have a bunch of different compounds that you would need to access. You'd need metabolites, genes, diseases, and symptoms if you wanted to answer this question. However, to do this, you would have to go to, for example, one repository like KEGG to get information about your metabolites. And you could go to UniPro to connect this up to the genes. And you could use OMIM to get the diseases, et cetera, et cetera, et cetera. And you notice that you have to hop between a bunch of these different repositories of information. These repositories of information, that's a long word to say, so I'm just going to call them knowledge sources in general. So these guys right here, these are our knowledge sources. It just contains a repository of all of our information. This is going to be a difficult task to do manually, right? So if you ask a student to do this, or if you want to sit down and do it yourself, you got to go to the website of KEG, you got to figure out how to work on their website or download the information itself, how to parse that information, and you finally get the metabolite that you want. Great, now you have to repeat the whole process for Unipro, et cetera, et cetera, et cetera. So it's difficult to be able to hop between these things. Let me know if you have any questions. I made this talk only last half an hour, so if there are no questions, we'll break early. But I do hear there is going to be ice cream afterwards, so maybe you don't want to ask questions that way. But in any case. Okay, so we got all these knowledge sources. We want to hop in between them. We want to be able to answer interesting questions by connecting these different knowledge sources. And there are a bunch of different interesting questions that can be asked. So for example, can we get all the diseases that have certain symptoms that affect other sorts of cell types? Or pathways that, when disturbed, lead to a malfunction in a certain organelle in a particular organ in people with a certain genotype. So these are really specific kinds of questions that might be interesting, especially for clinical applications. Or how can we find different drugs to treat certain individuals that have certain phenotypes or genotypes? So we want to answer these interesting questions. We want to use this information in the knowledge sources, but it's somewhat difficult to connect them together. So I guess that went away too quickly, but how? How are we going to be able to do this? So one effort is this thing called Translator. So Translator is basically a thing, project that is trying to integrate these different knowledge sources to be able to answer interesting biological questions. Translator is part of the national or project put on by the National Center for Advancing Translational Science, which is a mouthful. It's basically NCATS. It is one of the divisions of the NIH. So they basically took it upon themselves to have a large consortium project to be able to try to do this problem of integrating these different knowledge sources. The whole overarching project, by the way, if there's a reference right here, along with a link, if you want to get a like a 2-page general overview of what the Translator project is. But basically, we want to take all these different knowledge sources, all of these guys right over here, so you can see that these are a lot of the names that I was talking about earlier, the Mondo and the DOID, the KEG, everything along those lines. You take all these knowledge sources, and we want to be able to connect them up like that. So we want to be able to get them talking to each other very effectively. After they're talking to each other, we can then ingest these things and integrate them into some sort of system. So here you can see it integrated, and you can see that, oh, the genes and the gene functions and the pathways and the phenotypes and the diseases and the genotypes and everything like that all are in the same sort of data structure so that we can easily move back and forth between them with the purpose of being able to reason over it. Can we be able to connect up these little bits of information to reason over it in order to answer research-directed questions? With the whole goal of this thing being able to answer clinician questions. So if somebody comes in with a certain set of symptoms, maybe these are their, recorded on their EHR, and they have these sorts of genotypes. Can we then go throw it at one of these big integrated systems here to be able to get some sort of idea of how we might answer researcher-directed questions? That's going to be really annoying. Okay, a bit of historical context. This started in 2016 to 17, was connecting up all of these guys, whereas more recently is doing the actual reasoning as well as the question answering. Comments or questions so far? No questions at all. Perfectly clear, or clear as mud. All good? Okay, cool. Excellent. So nothing is done in isolation, of course. So this Translator project, part of NCATS, is a large team science thing. This is a small enough picture that it's not super useful, but it's just to give you an indication that there are many different teams involved with this. This is a subset of the different kinds of teams. And it's a very interesting project because while the grant application was, one, extremely non-traditional, but two, competitive, the actual performance of the grant, we are very collaborative, and all the teams are required to work together, share information, etc. So When I say we, I mean Team X-ray right up over here from OSU, Penn State, and the Institute for System Biology right here. Team X-ray, this is us. We are one of the reasoning teams that tries to do that question answering whole paradigm with some other teams from Stanford, Broad, UNC, UAB, et cetera, et cetera. So it's a very large kind of project. So that's what Translator is trying to do. How are they actually going to do this in practice? Well, I mentioned these knowledge graphs very briefly when I gave you the sort of overarching picture of what Translator is attempting to do, but let's go in a little bit more detail. So first off, how do we actually get these different knowledge sources to be able to talk to each other? So we need to be able to ingest that information. So we want to query those different knowledge sources, like pathway commons or KEGs, and be able to retrieve information from them effectively. So that's basically this part of the figure that I was showing on the page before. We want to connect up these different knowledge sources. So one way that you can do that is by using API queries. So I mentioned that these repositories are becoming a little bit less siloed. So thankfully, a lot of them have different API endpoints, which is basically a way for you to write a computer program to be able to then access it and pull information from it. Just in case if you might not know what an API thing is, I'm sure many of you do here do know. So what does that mean? That means that you pay some trainees and employees to do the enviable, non-enviable task of going through and writing a whole bunch of query Kimball, query OChem, query all these different knowledge sources and knowledge repositories. You basically just write little programs that allow you to talk to individual knowledge sources. And once you have a way of a whole bunch of ways to talk to a bunch of different knowledge sources, that will allow you to ingest these things, working on then integrating them together. Great. So integrating, I can now talk to my knowledge sources. I can pull things out of a knowledge source. Those different things that I can pull out, it might be a protein, could be a pathway, could be a drug. Those things, I'm just going to call them bioentities. It could be a particular SNP, it could be a genotype, I don't care, it's just a bioentity. It is a thing. These things are related to each other in some way, and so the first thing that you might think is, oh, I want bioentities and the relationships between them. That really sounds like a graph, right? Or a network, like nodes and edges, things and connections between them. So one very natural way to be able to integrate this information is using a knowledge graph, a graphical representation of that information. The thing that we're going to use is this really cool database called Neo4j. So Neo4j is one of these graph databases. It's not like a tabular database. It's not like an SQL sort of database. It is a graph database where literally the representation behind the scenes is a graph. So you have different nodes and you have different edges between them. The really cool thing about this is that it allows you a very nice query language to then be able to run any of your favorite graph algorithms. You want to do PageRank, that's built into Neo4j. You want to do a minimum spanning tree, there you go, it's already in here, etc. So it's very easy to run efficient graph algorithms in Neo4j. In fact, it's a pretty popular package. So you might every once in a while get alerts from your credit card company. It says, hey, did you make this purchase? Something like this. So it turns out that a number of credit card companies like Discover use Neo4j in the background for fraud detection purposes because they're adding your different purchases and the different locations and metadata about them into a big old graph so that they can run a graph algorithm and see if there's any sort of anomalous behavior, and they just send you an automated alert of, hey, did you make this purchase kind of thing. As well as product recommendation systems, so things like Amazon product recommendations utilize Neo4j mainly because you can handle tons of information in this thing. Which is great because our databases, as I mentioned earlier, are growing in size. So this Neo4j thing is going to be able to handle it all. So in our particular application, we started out and we ended up with a network that had 11 different kinds of bioentities, not 11 nodes, 11 different kinds. And you can see those bioentities listed up here. So we have things like molecular function, we have pathways, we have proteins, we have drugs, metabolites, biological processes, microRNAs, et cetera, et cetera. These are the kinds of things we have in this knowledge graph that were able to be connected from 14 different knowledge sources. So 14 of these different repositories of information, which ended up in about 128,000 nodes and about 7.5 million relationships between them. This was the first path, or first pass on this. We are now on the second iteration of this thing, and we have something like 5 million nodes. And is Finn here? How many edges was it? 160 million edges. Excellent. So a couple orders of magnitude, or an order of magnitude and a half larger. But this was the first approach that we did. So we got a big one of these. Nice thing is Neo4j can handle an arbitrarily large number of nodes, limited only by your disk space because everything is memory mapped. But then, after you've ingested and integrate, you want to ask questions of this, you want to inquire. So how can we use this to answer our interesting questions? Note, by the way, even if you're not doing one of these really large integration sorts of projects, Neo4j could still be useful to you. We all know in computational biology, networks and graph algorithms show up all over the place, as well as it's a great way to express information in a biological system. So Neo4j might be something that could be interesting to you to utilize in your project, no matter what your application area. Okay, so what about some early results? Let's see if we can't ask this an interesting question. So here's a question. Let's figure out what genetic conditions are protective against a disease. So usually genetic conditions cause diseases, right? But it could be the case that a particular genetic condition makes it such that it's less likely for you to get some other disease. So what we can do is we can sit down and we can encode a specific solution to answer this one exact question. So let's go ahead and, using a random walk, find genetic conditions in some sort of neighborhood near the particular input disease, which I'm calling X right here. And so let's look at a couple of instances of this. Let's say X is sickle cell anemia. What genetic conditions are protective? Oh, I think I actually have this backwards. Sickle cell anemia, malaria. Yeah, I have this backwards. But in any case, sickle cell anemia is protective against malaria. I had the X and the Y switched. But in any case, if you do a random walk against one of these diseases, in this particular case, that instance should be malaria. It turns out that sickle cell anemia is protective against it. And we can actually get that information from our knowledge graph. We see that, for instance, sickle cell anemia is that disease affects a particular gene, which is a member of a pathway. Another gene is a member of that pathway, which controls the expression of another gene, which is associated with a parent of malaria right here. So you can see that we've hopped through a bunch of different bioentities. And if we had to do this manually, that'd be really annoying to have to go to each one individually and to query all of these particular bits. But in any case, we can find that these particular genetic conditions are protective against these particular diseases, which, yes, this is sort of cherry-picked because these are ones that we actually know to be protective. So we were just trying to recapitulate known information. Question?
How do you get the sign if it's protective or increasing from this error?
Not very intelligently. We cross our fingers and hope, basically. This is, what's that? It could go the other way. Yeah, exactly. That's why this is an early result, because we're just trying to do a zeroth order approximation. Can we even possibly hope to get right answers in this case? Yeah.
I'm guessing that the initial network with all the different nodes branching off all the different nodes was not as tidy as this one, like each node would have.
Right. This is a particular subgraph of that massively tangled network.
How do you trim it down to this nice one that gives you the answer?
Right. So here, what we did is, using random walk, we took between every disease node, so given an input disease, we look at all the genetic conditions in a certain neighborhood, and we rank them based off of how close they are in terms of a random walk. And then we can say, great, so this one is closest, now just find me the shortest paths. Yep, and these are the two shortest paths from this guy to this guy and this guy to that guy. Yep. Did I see? Yeah, another hand.
So this isn't directional, is that right?
Correct. Yep. As I said, this is just sort of a zeroth order. Can we even hope to recapitulate known information from interesting questions? Yep. We will do something much more intelligent in a minute. Yep. Excellent. Good. So, as people are pointing out, this is a naive approach, but hey, it'll lend me a little bit of confidence that this might be fruitful to take this sort of approach. But I really want to generalize this and automate this and do this much more carefully. Because you don't want to have to sit down for every single one of these questions and ask things like, how do I make sure it's directed? And how am I actually going to answer this one specific question, etc., and then try to code up solutions for each one of the specific questions. Because, well, you could keep people employed doing that for quite a while. It's not going to be super efficient to generalize. So what we really want to do is we want to be able to learn on these graphs. And learn on these graphs in an automated fashion. So to be able to use your favorite machine learning algorithm, we are first off going to have to get our data in some sort of representation that will make it easier to learn on. So in particular, I want some sort of graph embedding. I want a vector representation of the nodes and edges inside of my graph. So let's say that we have this arbitrary graph. Here's a cartoon graph. And I want to be able to preprocess this so I can feed it into a machine learning algorithm. We might want to embed this thing using a certain encoding into a particular Euclidean space here. So you might imagine that these particular nodes sitting over here are embedded right over there, these blue ones, those red ones, etc. The whole point of this embedding or this encoding is that these individual points are then just vector representations. And with these vector representations, you can basically treat them as feature vectors and then throw it against your favorite machine learning algorithm and try to learn something about this particular graph right here from the actual vectors in the encoding. Yeah?
But how do you quantify the distance?
How do you quantify? Yeah, exactly. This is just sort of theoretically what embedding is, and then I'm going to talk about a specific kind of embedding in a minute. Yeah. Yeah, but you're thinking exactly along the right lines. Yep. So there are many different methods to do this, right? So there's one node to vec, which is based off a popular other sort of vector representation called word to vec, deep walk, line, graph rep, hope, Laplacian eigenmaps, graph factorization, etc., etc. The one that we're going to use is this very first one that I mentioned here, node to vec. So node to vec here is going to be the one that I'm talking about to actually concretely describe how we go from these particular nodes and edges in the graph down to the actual encoding. So what does node to vec do? It is another random walk based Euclidean embedding technique for graphs. So here's the reference for this particular embedding, but the whole basic idea is as follows. We want to figure out exactly what you asked. So what is the distance? How do we actually encode these things? So let's say that we have a particular node like U sitting right over here, and another node V sitting right over here, and these are keeping track of their encodings. So over here I have the encoding of U and the encoding of V. So I'm going to describe how to go from those individual nodes to the actual individual encodings. So the approach is that we want to optimize it so that the similarity of the encodings, that is the similarity of the vector representations, which here is represented by kind of the angle between them. So if you look at this, this is the dot product between the encodings. This right here says similarity. So similarity between the encodings is defined to be the dot product between these guys. We want that similarity to approximately represent something about the similarity of the nodes in the graph. So in other words, if two nodes are basically completely identical in the graph, we want their encodings to be very close to each other. If two nodes are extremely different in the graph, we want their embeddings to be basically perpendicular to each other. Great. So how do you actually do this? we want this graph similarity. We're going to define that as the probability that a random walk starting at one of the nodes hits one of the other nodes. So let's say that you start at V, and if you had a high probability of hitting U, then you would want your encodings to be similar. That makes sense? Kind of work. Then you have to actually define what you mean about the probability of a random walk starting at one hitting another. So we can formalize it and represent this as a probability of seeing U given that we started at V. And then we use a kind of softmax representation of this particular probability. So you can see that we have utilized our actual dot product in there because indeed we want these things to be similar, so they're going to have to be related to each other. And that's how we're going to define our probability. So if we have our encodings are similar, very similar together, we should have a high probability of running into one of the nodes starting at another. Great. How do you do this in practice? You are going to generate a neighborhood around each one of the vertices in your network. And then you're going to maximize the probability of observing that particular neighborhood conditioned on the encoding that you picked. So what does this mean? It basically just says you pick an encoding. You look at a neighborhood. You say, what's the probability of that neighborhood given the encoding that I was talking about? If it's low, well, tweak your encoding. Do it again. Calculate the probability, et cetera, et cetera, et cetera. And you'll end up with something that looks like this. Maximize over all of your different encodings the log probability of observing that neighborhood. So conceptually, if nodes in this particular graph right here look like they have similar neighborhoods, they are going to be grouped similarly together in node2vec. So while this particular picture that I generated right here is completely a cartoon, and I completely arbitrarily generated this data, it does sort of recapitulate the information. So right here, all these nodes are kind of similar because they have similar looking neighborhoods to each other. So node2vec is going to group them together. Whereas, say, this blue node over here and this green node over here have very different looking neighborhoods, and so they are going to be graphed very far away from each other, like this. So that's node2vec. Fantastic. Of course, with any one of these, there are a few different knobs to fiddle. There's one, exploratory parameter P, which is when you do your random walk, how likely are you to step out or just stay where you were at an original node? And do you want to explore in sort of a breadth-first search or a depth-first search fashion? Do you want to go around or do you want to go out, basically? But this has been used quite a bit in computational biology. So if you're trying to get into machine learning in computational biology, this can be a great way to represent graphical information. It's been used things for protein-protein interaction networks, using Parkinson's disease gene predictions, pathway reconstructions, essential protein identification, a bunch of other things. But in this talk, I'm going to talk about how we can use it to try to do drug repurposing. Great. These slides will also be online soon, so you can get the links. So, drug repurposing. What is the problem? Drugs are expensive to develop. So we have drug companies that are spending millions of dollars, and most drugs fail through the clinical trials. So if you are funded by the government and you are just a division of the NIH and you're like the NCATS and your whole goal is to treat rare diseases, and there's not a whole lot of money in rare diseases, one thing that might be more fruitful is can we use existing drugs that are already FDA approved and just find new uses for them? Because we already know that it went through clinical trials, they're already approved, we just find a different purpose for the drugs. So that is drug repurposing in a nutshell. So let's go ahead and use that previous knowledge graph that I talked about in the beginning, the one with hundreds of thousands of nodes and millions of edges, and throw it through node2vec. So we had our bunch of different repositories of information. We had 14 of them. We had our big old knowledge graph and all of the different bioentities inside them with 100,000 plus bioentities and a bunch of relationships. We're going to throw it through node2vec. So every single one of the nodes in our knowledge graph is then going to be embedded as a big old vector right here. Then, to use this for drug repurposing, I'm going to need some training data of known drugs which treat diseases, right? So I can use a couple other repositories of information, SemMedDB, which is really interesting. They use natural language processing to look at all the different abstracts in PubMed and then pull out relevant terms. MyChem, which is another curated repository, and then actually this NDF that is sitting right here, this is the National Drug File. This is basically the big old file put on by the Veterans Administration that tells doctors which drugs are okay to prescribe for which diseases. It's like, okay, this will be useful for training data. Because basically, we get from this 80,000 plus drug treats disease pairs. This drug treats this disease, as well as 31,000 this drug should not be used to treat this disease. We got to be a little bit careful here because, right, if we're saying this drug treats or it does not treat, that's very different than saying this drug treats and this drug will actually make the disease worse, right? So you can see how there's slightly different problems, and depending on the training data that you use, they could be slightly different. But in any case, we'll address that a little bit more in a second. I'd be remiss without saying this is with a student and a postdoc at Oregon State University working on this particular project. Great. So how are we going to do this learning? In the end, we're going to end up using a random forest. Not going to go into detail of a random forest, but it's like a ensemble decision tree sort of approach. But the whole basic idea is that after we have these feature vectors, one for each node in my graph, I can then take an encoding for a drug, an encoding for a disease, and concatenate them together. So if I know this drug treats this disease, I look at their vector representation, I put them together. This is now a big old feature vector for drug treats disease example. And I take this drug, and my training data says it does not treat this disease. I take the vector representation of that one and that one, I concatenate them together, and that's a vector representation of this drug does not treat this disease, etc. I do this for all my training data. And then I can throw it inside a random forest, like right over here. And we found out that it was optimal when we had about 2,000 trees of max depth of 32. But in just a minute, I'll give you a little bit more about the accuracy here. Basically, the way we train this thing is we want to maximize our AUC, given our training data, our true positive, and our true negative. True negative, not false positive. Training data using tenfold cross-validation. Okay, so results. First off, remember how I said that we got to be careful because our training data, it's different saying drug does not treat disease or drug should not be used to treat disease, and how those are slightly different. So this is where we need to consider this. Our training data is very biased. In particular, we have a massive class imbalance problem. You give me a random drug, you give me a random disease, with overwhelming probability that drug is not going to treat that disease, right? You give me some, otherwise we'd all be out of business, and where doctors would be, et cetera, because you just keep throwing drugs at it and eventually it'll work. So that does not work. So we think that with overwhelming probability, a randomly selected drug and disease pair will not actually work. And so we might not be able to use a very simple machine learning algorithm, like logistic regression. So if we tried to use logistic regression, like I have right here, and I look at the rate of positive prediction versus the cutoff probability. So for instance, logistic regression will return me back a scalar between 0 and 1. If I say that number, if it's above a 0.8, I say it treats. And then we look at the fraction of positive predictions. Here we get a graph that looks like this. What does this mean? The blue are my true negatives. I want this graph to decay really quickly, because that means I'm effectively learning that these are really true negative cases. This green guy right up here, that's my true positives. I want this thing to be all the way up over here, because that means I'm going to be effectively learning that these are true examples. What I'm going to do with these random pairs, this is when I'm using random pairs of drugs and diseases. Well, I just said a random pair with overwhelming probability should be a doesn't treat, right? So the rate of positive prediction, we would want that to be low down here. This is not low, right? this is bad. Because a random pair looks basically like a true positive. That's not very good at all. So even though, in general, I always recommend whenever you're trying to do machine learning, start out very simple and then build up and only get as complicated as you need to solve the problem at hand. Here, very simplest logistic regression, not really going to work because of our class imbalance problem. So let's instead move to a random forest. You could use a random forest of a shallow depth. This is even worse, right? So here our true positives are actually dwarfed by the random pairs. But now let's increase the depth of this thing and make it a deeper and deeper and deeper random forest. And oh, now this is actually starting to look like how we want it. We have our blue line decaying quickly, our green line is almost all the way to the top, and our random pairs right here, at least there exists a nice little sweet spot in here. So if we set our cutoff probability that says if our random forest returns a value greater than 0.8, with high probability, it'll correctly get the true positives, and only a small fraction of random pairs will be predicted to treat. Does that work? Fantastic. Good. Then, using this particular depth of a tree, depth 32, we can look at the area under the curve, look at a couple of other performance metrics, and here we can see that we get an AUC of about a 0.93, and you can look at the F1 score there, which basically says that, yeah, we are actually learning something about the drug and the diseases being treated here. So let's go ahead and see if we can't look at a couple of predictions here. So let's first off see if there are any recent drug and disease pairs that are known to treat that weren't in our training data, but are already sort of established and known. So what Finn did in the back there, he went through the very tedious task of looking at a whole bunch of these different treat probabilities and then using yet another knowledge source to be able to say, were these actually known and it was just missing from my knowledge graph? And we came up with a whole bunch of drug and disease pairs right here, where there was actually known publications that said, hey, this drug can be used to treat this disease, but they just happen to be absent from our training data. This is really good news, right? Because these are nice ones that are sort of out of sample predictions that turns out to have been supported in the literature right here. And some of these are actually pretty cool. So dexamethasone being used to treat for a certain eye disease, posterior uveitis. This one, the reason why it wasn't in our training data is it's only in a phase 2 clinical trial. So it's not yet completely approved, but we thought that was particularly interesting. So don't think it's, it could be possible that we can get even better performance gain by going from a random forest to something like a neural network, et cetera. But at least even this very simple approach sort of confirms that using this knowledge graph representation, this vector embedding and learning, you can actually get something interesting out of it. So, in conclusion, knowledge graphs, you can get really interesting results from integrating these different public repositories of information. These graph embeddings like Node2Vec or Deep Walk or these various things that I mentioned are great ways to learn features on large graphs. If you're doing computational biology, I'm pretty sure you ran into a graph at some point, probably a big one. Drug repurposing is possible by combining these different ideas here. And very lastly, a shameless plug. This is not the only thing that Translator is trying to do. Translator is actually trying to make basically a biomedical Siri, so you can ask questions of one of these automated systems. I hesitate to give you guys the URL directly because I don't want you all to like hit it at the same time and have the whole system just come crashing down. So I'll give you a couple of screenshots, but basically this is our team's sort of implementation. You can go and you can ask it a question right up here with a nice little autocomplete guy, and it'll sort of give you a graphical representation of what your query might look like, and then after that return you a couple of predictions and sort of supporting information and confidences and things as well. So if you're really interested in this, I can probably give you the URL individually. That way you can slow down the number of requests. But in any case, I want to thank my collaborators, the co-PIs on this grant, Steve Ramsey from Oregon State and Eric Deutsch from Institute for Systems Biology. couple other ISB team members, OSU team members, as well as a UW, another scientist who's actually studying us, scientists studying this project, but he's a sociologist. But in any case, the actual drug repurposing, as I mentioned, a couple of trainees at OSU, Finn Womack, who's actually here, as well as Jason McClelland, acknowledging funding. Maybe during the happy hour, if you're interested in to hear about how this funding thing happened, you should ask me, because you'll notice right here, this is an OT. You might not recognize an OT grant. This is an other transaction kind of grant. So no, we didn't actually just have to write a proposal. We actually had to solve some ciphers and just do some puzzles. So if you're interested in hearing about this, ask me about it. But in any case, I'll be happy to take any questions if you have any.